For practical, the thing I wanted to tell you all about is about the blood grouping system, or how can you find out how what is the blood group of a person. So, as a part of immunology, blood is a very important part in this particular subject, immunology. So that is why the blood grouping system is also an important part. Now we have already studied about transfusion, blood transfusion, and then we studied on why all type of blood groups cannot uh, be transferred to other person. That is how hypersensitivity takes place because of wrong blood transfused to a person. Okay, now let us see what is this blood group system. Okay, now blood grouping system was first discovered by an Austrian scientist whose name was Karl Landsteiner in the year 1900. The blood group system that he discovered was ABO blood group system. So he had performed an experiment long back. He mixed different blood types, and then he found that the plasma from certain blood type produced agglutinates or formed clusters, which were caused by the absence of molecules on red blood cells, and resulting in antibodies to defeat that molecule. That means when this blood he had mixed different types of blood groups all together, then he found that the blood groups agglutinated. Agglutinated means, you know, those that started clumping. Okay, all of them started clumping. They got separated. Those lyses of blood they took place, and then they all started clumping. That is called agglutination. And then this was a discovery for him, which let him find out that actually though everybody has got blood, but the blood of every individual is not same. There are certain blood groups in which this can be differentiated into. Okay, so this particular discovery has awarded him the Nobel Prize. Now then, this blood grouping system plays a very important role in blood transfusion. Obviously, if we would not have known about the blood grouping system, blood transfusion would not have been possible. Now, our immune system, it recognizes another blood type as foreign. If I have B positive blood, the blood that has to be transfused inside me has to be B positive. If some other blood group is transfused inside me, then that will be considered to be a foreign blood. Okay, foreign blood group. Even sometimes what happens, even same blood group also sometimes creates problem if the person is hypersensitive, and then that blood group may cause itching in that person. So immediately the blood transfusion is stopped. So it is happening because the blood that is that is being given to you or transfused within you is a foreign blood group. It's not your own. Okay, so any inappropriate match with the RH and the ABO blood type causes the most serious and life-threatening transfusion reaction. Therefore, before blood transfusion, it is suggested to have a blood group check. So this is about the blood grouping system. Now, in this blood grouping system, there is another system. The blood grouping is the ABO system, and the another system which is within this is the research system. Okay, which is the RH. Now, what is this RH factor in the blood? We know that the blood groups are A, B, and O. These are the different blood groups. Okay. Now the research factor is the when we classify these blood groups as AB positive, AB negative, B positive, B negative. The positive and negative is a research factor. That is considered as a research factor. Now the ABO blood group system consists of 4 types of blood group. A, B, AB, and O. And it is mainly based on what? It is mainly based on the antigens and the antibodies on the red blood cells and in the plasma. Both antigen and antibodies are protein molecules in which the antigens are present on the surface of the red blood cells and antibodies are present in the plasma, which is involved in defending mechanisms. So what do we understand here? That in one particular blood, you will find the presence of both antigen and antibodies. The antibodies will be for yourself and the antigens will be for others. Are you understanding? What does it mean? I'm telling that antibodies are for yourself means if some a blood which is not compatible for you, a blood group which is not compatible for you is entering into your body. So that is what? That's an antigen. So the antibody that will be present in your blood will fight against the antigen that will be present in that other blood group. So that is why both the antigens and antibodies will be present. Now, if your blood is transfused to another person who is not of the same blood type like yours, so the antigen that is present in your body, in your blood, will be affecting the other person, and his antibodies will be fighting with the antigens present in your blood group. So that is what is the why these antigen antibodies are present in the blood. Now, on the other hand, the Rh blood group system consists of 50 defined blood group antigens. See, there are different types of antigens present in this blood, almost 50 defined blood group antigens. In the Rh system, the most important antigens are the D, C, small c, capital E, and small e. Now, let us study about this blood group system. The basis of grouping is of two antigens, antigen A and antigen B. The ABO grouping system is classified into 4 types based on the presence or absence of the antigens on the red blood cell surface and the plasma antibodies. Okay, now here what did we find? That this antigens, they are present on the surface of the red blood cells, okay, and the antibodies which are present are in the plasma. The blood has got different layers, and the layer which has the plasma of the blood consists the antibodies in it. Okay, now as we are talking about the different blood groups. Now, what does group A have? Now, if you say blood group a blood group A will have the antigen A, okay, and antibody B. Please remember this. It will have antigen A and antibody B. Blood group B will have antigen B and antibody A. Then blood group AB will contain both A and B antigen, and it will have no antibodies, neither A nor B. And blood group O will have none of the antigen, nor A or B antigen, but it will have both the antibodies A and B. I hope this part is clear to you all. Now, moving forward, the ABO blood group system is important during blood donation or blood transfusion, as mismatching of blood group can lead to clumping of the red blood cells with various disorders. It is important for the blood cells to match while transfusing, that is, donor-recipient compatibility is necessary. For example, a person of blood group A can receive blood either from group A or O, as there are no antibodies for for A and O in blood group A. Okay, so here you see, I had told that blood group A contains antigen A and it has antibody B. Since it contains antigen A, so if we require blood for group A, then we will have to add only group A blood. You cannot add a blood group B for this, okay, because if you add blood group B here, the antibody B will act against the blood group B. Fine. Similarly, it's a vice versa. Now here they are telling that a person with blood group A can receive blood either from group A or O. Now, why O is allowed to be given to a person with a blood group A? Because there are no antigens in A. In blood group O. If there are no antigen in blood group O, so if that blood group is received by a person with blood group A, so the antibodies present in A will not react with anything because it has no antigen. Okay, and antibodies can do nothing. Because it has a B antibody and this also has B antibody, so it will not hamper. But since it has no antigen in it, therefore even after addition to group A, it is going to cause no harm because this the blood has no antigen in it and the antibody present in it will not react with it. So that is why blood group O is like a universal donor. Okay, so here you see this is a table of the blood grouping system, ABO blood grouping system. This is group A, B, and O. Now, here in blood group A, we have the antibodies anti-B. In blood group B, we have antibodies anti-A. In AB, there are no antibodies. In blood group O, there are two antibodies, anti-A and anti-B. And coming to antigen, A will have A antigen, B will have B antigen, AB will have A and B antigens both, and O will have no antigen. Okay, so this is how the blood grouping system is. So in addition to this blood grouping system, another system we need to know is the RH blood grouping system. In addition to the this one, ABO blood grouping system. Why is this RH blood grouping system important? Because ABO blood grouping system is, this RH blood grouping system is important because there are many people having different RH factor. Now, about 2/3 of the population contains the third antigen on the surface of the red blood cells known as RH factor or RH antigen. This decides whether the blood group is positive or negative. If the RH factor is present in an individual, then that person is considered as RH positive, rhesus positive. And if an RH factor is absent in an individual, that person is considered as RH negative, as they produce these RH antibodies. Therefore, in when we see the compatibility between donor and individual, this is another big factor that has to be kept in mind, the RH factor, and that will what do you say, that will help you to know whether the person is actually a particular group positive or negative. When we say A positive, or A negative, B positive or B negative, the positive and negative depends upon this RH blood group system. So we also need to find out whether the person has this RH antigen in its body or not. I hope this particular thing is clear to you. Okay, in order to perform the blood grouping test, you don't require any big instrument or something like that. What we require is a kit which has all the antigens present in it, and we need to add these antigens to that particular blood group, to that particular blood drop that we take from the person in order to identify the blood group. Okay, we need the sample, that is a drop of blood from the a drop of blood is put on the slide, is and the rest of the things that are required is the kit which has the antigens that are required to be added to those drop of blood. Now with this video you'll get to know how it is done.
ABO blood group system and RH blood group system in previous video lectures. We also saw that on the basis of these two blood group systems, humans have 8 blood types. In today's video lecture, we will explain blood typing or blood group test. Blood typing refers to the test to determine the blood group of a person. This test is based on agglutination reaction. Recall that agglutination reaction is an antigen-antibody reaction in which antibodies cross-link particulate antigens resulting in the visible clumping of particles. Agglutination of red blood cells is known as hemagglutination. In Greek, hema means blood. So, hemagglutination is the clumping of red blood cells. The target antigens in this case are present on red blood cells. When specific antibodies to these antigens are added, the antigen-antibody reaction results in agglutination, which is visible as red granules. Suppose we have an individual with blood group A. That means his RBCs have A antigens on their cell membrane. We put few drops of blood from this person on a glass slide and then add anti-A antibodies. We will mix them. After some time, red granules are visible like this. These granules are the result of clumping of RBCs. Let's now understand how blood typing is done.
Good grammar and spelling are important, but if you want to write essays that inspire, messages.
For this test, we will require few drops of blood from a person whose blood group is to be determined. We know that blood will contain the target antigens on RBCs. These target antigens are A antigen, B antigen and Rh antigen. We will detect the presence and absence of these antigens in blood and determine the blood type. Next, we will need antibodies specific to these red blood cell antigens. So, here we have 3 reagents labeled anti-A, anti-B and anti-Rh. Reagent labeled anti-A contain antibodies against A antigens. Similarly, anti-B reagent contain antibodies against B antigens. Reagent labeled anti-Rh contain antibodies against Rh antigen. We will take a clean glass slide and mark 3 circles. Label them as anti-A, anti-B and anti-Rh. We will put a drop of each reagent in the respective labeled circle. Next, we will add 1 to 2 drops of blood in each circle. We will mix the reagent in the blood and observe the slide after some time. Agglutination, that is, visible red granules, will confirm the presence of antigens. No agglutination means the antigen specific to the target antibody is absent. Let's look at the possible observations and their interpretations. Here you can see agglutination in anti-A circle and anti-Rh circle. This means antigens present on red blood cells are A antigen and Rh antigen. No agglutination in anti-B circle. Therefore, B antigen is absent. So, blood group is A positive. Recall that plus sign indicates the presence of Rh antigen. Here, agglutination is observed only in anti-A circle. This means antigen A is present on RBCs. No agglutination in anti-B and anti-Rh circle. So, B antigen and Rh antigen are absent. Therefore, blood group will be A negative. Recall that minus sign indicates that Rh antigen is absent. Here, no agglutination in anti-A circle. So, antigen A is absent. B and Rh antigens are present since agglutination is observed in anti-B and anti-Rh circles. So, blood group will be B positive. Here, antigen A and Rh antigens are absent since there is no agglutination in anti-A and anti-Rh circles. Agglutination in anti-B circle indicate presence of B antigen. So, blood group is B negative. Here, agglutination is observed in all 3 circles. This means all 3 antigens are present. Therefore, blood group is AB positive. Here, agglutination in anti-A and anti-B circle indicates that both antigen A and B are present. But there is no agglutination in anti-Rh circle, which indicates absence of Rh antigen. Therefore, blood group is AB negative. Here, No agglutination is observed in anti-A and anti-B circle, therefore both A and B antigens are absent. Agglutination in anti-RH circle indicates presence of RH antigen. Therefore, blood group is O positive. Here, agglutination is absent in all the circles. This means all the 3 antigens are absent on the RBCs. Therefore, blood group will be O negative. Blood typing is used to determine the blood group of donor and recipient prior to transfusion. Also, it is used to determine blood group of mother-to-be. Why blood typing in these cases is important? What are the consequences of non-compatible blood transfusion? All these questions will be answered in the next video lecture.